Okay, in my face-to-face class, I introduced a little short idea that I thought was important for our discussion. And so I wanted to make just a short video to kind of add on to the material that we're talking about as far as DNA structure. So basically, we're going to take a short side trip here. Now, as you remember, under Chargaff, we were talking about the fact that he was really testing this idea that Levine had put forward about 1910, that DNA was in some kind of regular structure with the bases all being part of that structure. And it's sort of a quadrilateral form, right? So they called this the Tetranucleotide Hypothesis. And the actual prediction from that was that the amount of A and C and G and T would all be equal. And Levine disproved that. He said that that's not, when you found certain types of DNA, particularly humans, fruit flies, more advanced forms, the ratios were not equal. And so this showed that there really were other ways. It wasn't built in this tetrahedral form. But it turns out that there are some times when there are tetrahedral forms in DNA, and I wanted to talk about that a little bit. In 2013, a paper was published that outlined some very nice techniques in order to look at these structures. What they developed were some antibodies, and what those antibodies were to are what are known as Quadruplex structures. And what they amount to is this. They amount to a series of 4 Gs that are interlinked, usually with some molecule in the middle, potassium in this case, and then some ligands, some structures that connect them together in almost like a ladder-like form. And these occur naturally inside of our DNA in certain places. And these individuals developed an antibody. Now this idea had been around for since the early 2000s, but it took about 12, 13 years to develop some proper antibodies. And what those antibodies, as you can see in these cells, these red spots, are places where these types of structures, these G structures exist. And then here's a set of replicated chromosomes in some kind of condensed state. And here are these red dots indicate that in these chromosomes themselves, you see these structures. Now these structures are held together in quite an unusual way. The bases themselves, this is again DNA, so the bases are not in the typical Watson-Crick bonding angle. They are in slightly different form known as the Hugstein angle. This was a series that was discovered about 10 years after Watson and Crick published their information. There was another form of a naturally occurring DNA connection that was found out. Now notice what you see here is on the bases in this purine, you're linked up here in your typical form here, but you've changed that to where now both of the rings are involved in the connection in the hostine. So it's a different sort of a flipped form of that. Okay. Now, what's interesting here is that these quadruplexes are known to interfere with telomerase. Telomerase is the enzyme, and we'll talk about that in the replication lecture, but it's the enzyme that fills in the ends of the chromosomes. Okay. Now, what it does is it forms this sort of structure that literally causes telomerase not to work very well. Now, what is interesting about it is that telomerase is highly active in the replication phase, synthesis phase, in cancer cells. Remember, cancer cells are cells that are just replicating sort of uncontrollably. And to do that replication, of course, they have to go through DNA synthesis. And telomerase is highly active trying to set those up. So some individuals said, well, you know, if we can somehow, if there's a naturally occurring part of DNA that on occasion interferes with telomerase, and we can cause that to occur, maybe we can slow down these cancer cells long enough for other therapeutic agents to take hold. So telominostatin was produced. It's an anti-cancer drug. It's a fairly complicated drug to make, but it's an anti-cancer drug. And what it does, it induces these tetraplexes to form. Now it turns out that all these tetraplexes don't have to be guanine based. They can actually be some other forms that we found out, but they are often guanine based with loops and things. And what they do is when you put the drug in, they induce this sort of tetraplex formation with these ligands holding together. When you have telomerase coming in, then it doesn't work. It can't get past these structures. Now, why does this matter? Well, it matters because what's known as the Hayflick limit, and the Hayflick limit is the idea, it's a hypothesis that the number of times that cell can divide is limited. We thought up until the 1960s that cells were immortal. They could just keep going through the replication process, but there is a limit. Most human cells are about 40 times that they can go through replication before they start to senesce and die off and have to be replaced. Some can go 80, just depends on the things. Now we can immortalize cells. We can take cells, embryonic cells, and we can immortalize them. That's what, or even non-embryonic cells, the HLA cells, or breast cancer cells that have been immortalized. We can do that, but cells in the body have a limit to the amount of times. Now what drives that limit is the shortening of the telomeres. And once they get to a certain short length, the cell starts to senesce and eventually will die. That's known as the Hayflick limit. So the idea here is that you put this drug in, you've got the most rapidly cells that are expanding are cancer cells. The telomeres in those cancer cells start to decrease, and with that you then start to have those cells start to senesce and slow down long enough for other agents to come into play. So it's quite an interesting idea. Now the problem you have in actually using it is that if you want to put it in clinical trials, by the time people get to clinical trials, a lot of the replication has already gone on, and this takes a while, right? Because you have to, it needs to be there for quite some time. Well, they don't have that much time to work. So getting it there, getting it to the locations and that sort of thing is one of the leading ideas right at the moment. It turns out there's quite a few different ways that you can mess with telomerase, if you want to use the word mess with, that you can cause it to quit working. And this is just one of the therapeutics that's being involved there. All right, so just a quick video on how this works, and just wanted to kind of show you what we had talked about in lecture.